There we go. Hello, everyone. This is Cancer Genomics Data Pipelines. I'm Lynn Langit.
And I'm Samantha Langit.
And we're going to be presenting information from CISRO Bioinformatics Australia. Samantha's first going to start by talking a little bit about the problem that we've been working on.
First, let's talk about DNA. There are around 3 billion DNA letters or nucleotides in one person's genome. That's 3 billion data points per person. And with up to 25% of the population possibly getting sequenced by 2025, that's a lot of data that we need to be able to process.
Samantha, why is it that sequencing is becoming so much more accessible?
Well, with increased computational capability, this drives down both the time it takes and the cost of getting sequenced, making it much more accessible to greater portions of the population.
And it's really, in many ways, the ultimate big data problem. So to introduce ourselves, I'm going to introduce first Samantha Langit. Samantha is my daughter. She is 18. She has just graduated from high school in the United States, and she has intended to go to college to be a bioinformatician. Samantha's career in bioinformatics, however, started last summer when, during the summer, she had a great opportunity and she was able to attend Stanford University through a special program for students who are interested in these fields. During that time, one of her courses that she took was cancer bioinformatics. And because of one of the comments that she made after studying at Stanford and coming back home, I decided in my profession to be interested in this. Her comment was, gosh, they're not using the cloud and cloud data pipeline patterns as much as I would have expected. So Samantha's going to represent the bioinformatics position in this talk, and then Samantha, I guess, is going to talk about me.
This is my mom, Lynn Langit. She's a cloud architect. She's worked with Google, Amazon, and Microsoft Cloud. And she's going to be talking about the speed, the cost, and the simplicity of each possible big data solutions to some of the genomic problems that researchers face in this area.
So because I know that this is a technology conference and not a biology conference, what I wanted to do is I wanted to take patterns around big data pipelines that I've been observing and working on with the bioinformatics community and generalize them so that they could be of use to a larger audience. As I've done work in big data domains from advertising, education, IoT, and now bioinformatics at a very huge scale in the public cloud, I've found a series of patterns that are transferable. So I'm hoping that you'll get that out of this talk. To start, I've created this pipeline process that I use in building these high-performance data pipelines. It has 5 parts to it. The first part is to clearly define the business problem. That's kind of standard stuff. But I do find when there's huge data volumes that there's this tendency of using the latest and greatest technology really without regard sometimes to matching it to the business problem. So you got to start there. So we're going to talk about 2 bioinformatics problems in this talk and show you pipeline solutions. Then we need to look at not only the quality and the quantity of the data, but the type of data. Genomics is really interesting in that the data has some unique characteristics that Samantha's going to talk about. The third phase of the pipeline is probably of the most interest to this audience. It's the process by which, as an architect, in working with this community, I've helped the bioinformaticians to identify the candidate technologies for the common parts of the pipeline, the ingest, the extract, transform, and load, or extract, load, and transform, the business analytics, the machine learning, and the visualization. Now, I'm a big proponent of lean startup or lean type of processes, and I apply this to data pipelining in that we build MVPs or portions of the sections via the candidate technologies. We iterate, learn, and then we assemble them and then test at scale. So in both of the problems that we look at, we're going to discuss from an architectural point how some of the decisions were made and some of the general patterns I'm seeing in these huge data pipelines. So to get us in the right mindset, my background is traditional data warehousing, actually even DBA. For some of you who've followed my work for a while, I used to work at Microsoft 7 years ago, was a very big user of SQL Server, wrote 3 books on analysis services. So I have this background in traditional data warehousing that one of the reasons I come and do these talks is I've made this sort of transition to broadening my tool set, including these types of traditional solutions for those types of problems, but also building new types of solutions for problems of the scale of genomics. And I would tell you that one of the first things, if you've worked with this sort of a solution, that I would recommend that you try to do is literally put it out of your mind. Because none of this architecture do I find fits in these big data pipeline solutions. You just basically have to start with something totally fresh. In looking at these problems, I worked with some bioinformaticians, and I found two types of problems that we have pipeline solutions that we're going to talk about today. And Samantha's going to talk from a bioinformatics point about the first problem that we worked with.
The first problem is genomic sequencing results. Now, recently in molecular engineering, there's been a huge breakthrough in the development of a technology called CRISPR-Cas9. This allows researchers and medical workers to edit the genome at the level of almost to the specificity of specific DNA letters. Now, the NIH in the United States has approved this for human health research, and it could revolutionize the way that we treat cancer. However, in order to find the exact possible great targets for CRISPR to edit, we need to look through a huge amount of data, those 3 billion data points that we mentioned earlier, and find very specific results.
So when I heard about this problem, I thought, this sounds like the kind of problem for which the serverless architecture that we've heard so much about this conference would be a perfect fit. Now, I happen to do the majority of my work on the Amazon cloud because that's where my customer demand is, coming from the West Coast of the United States. But this pattern is, of course, implementable in other cloud vendors such as Google or Azure as well. Just this is the base architecture that I was expecting to work with bioinformaticians and to design and build to address the problem that Samantha talked about. So if you're not familiar with it, you start at the bottom level with the data tier with S3 and usually DynamoDB, and then at the compute tier you have Lambda functions and you interface with the rest of the world with API Gateway. Now this is a simplification. You'll often have machine learning and other services associated, but just in case you're new to this architecture, this is what I was expecting to work with bioinformaticians to build as a solution to this particular problem. Now, speaking of bioinformaticians, I made a trip this year out to Australia for another reason. And while I was there, I had the good fortune to be invited to come and speak at CISRO. You may not be familiar, I wasn't. This is similar in Australia to, in the US, the National Science Foundation. It's a national science research organization, not only in Australia, but in that section of the world. So it's the Commonwealth Scientific and Industrial Research Organization. They have a very large bioinformatics group that's working on solutions to bioinformatics problems for humans, animals, plants, so on and so forth. So they're based in Sydney. And this is a small section of the group that I worked with. The woman there is Dr. Dennis Bauer. She's the lead for the group, and there are two of her people that she works with. Now, interestingly, they were working on the CRISPR-Cas9 problem, and they actually came up with a solution. And that solution is called GT-Scan2, and Samantha is going to take me through a demo of this for you now. So what are we looking at here, Samantha?
This is what you see when you first see the site for GT-Scan2. It's a search querying database to be able to find the best possible candidate targets of DNA sequences that CRISPR can edit. So let's look at what happens when we click scan. So right here, we're going to look at the genome that we have available for use. The chromosome, we can specify that. And we can also specify, if we don't know the exact letters, but we know the placement of them, we can specify the place in the genome that we're looking for. Let's load up an example.
So Samantha, when this comes up, is this all that the researcher is going to need so that they can find that place as a potential target for editing, for experimentation? Is this all they need?
Not necessarily. Even in this sequence itself, there could be areas that CRISPR might trip up on while editing, so we need to find exact sections that would be most useful for what we're trying to prove. To do that, let's look at example.
And in the example here, what are we looking at? Can you explain this screen to us a little bit more?
This would work kind of like a Google search might work. up at the top in the query box, we were having the section of DNA that we're looking for, that we specified when we said scan. Now, on the bottom here, we have a list of results, organized from top to bottom in relation to their relevance. For example, this top result right here, or that specific result right there that my mom just clicked on, that's highlighted down below. It has high activity, which is good, something that CRISPR looks for, and relatively few off-targets, which is also something that CRISPR needs.
And so if you click on a black, what does that show you?
That shows you a segment with low activity. That probably means that that DNA is either bunched up or not in use. So it wouldn't be a good idea to hook the mechanism on and try to edit from there.
So Samantha, we're just looking at a small portion of the 3 billion data points here. I see from 295, whatever it is, to 295. So we might have to sort of dynamically search the genome results, kind of like you said, like searching Google, to find potential targets for editing. That's how this tool is designed to be used. Is that correct?
That's correct. It compares this result to a number of reference genomes that it has in a database to find the best possible target.
Great, cool. Okay, so like I said, I was expecting, because when I worked with bioinformaticians previous to the group at CISRO, I found that many of them had big gaps in their knowledge, and so they were trying to do processing on local clusters, even desktops, maybe using EC2 instances. I certainly didn't find anybody building a serverless pattern that was scalable for this kind of a bursty workload. That was until I met these people at CISRO. The way I met them was the underlying architecture that they built for GTScan2 was actually serverless. So one of the reasons that I open with this is because, I'll admit it, a lot of us work on what I call silly problems. I live in Los Angeles, so I've worked on Snapchat remonetizers, Instagram, reservers, YouTube remonetizers. And one of the reasons I've decided to give talks about this topic is because Dr. Bauer and her group have 3 open recs. They cannot hire people who are qualified to help them with this architecture. It's very, very interesting. So I'm talking about this worldwide. In fact, if you want to move to Australia, they will relocate people worldwide, or anybody watching on the video, who is familiar with a serverless architecture, because for this type of a problem, they have gotten an extremely good result. And in fact, they have won awards around the world for this architecture. It is their intention to place this tool into the public domain for bioinformatics research worldwide. Now, my interface with them was because I'm an Amazon community hero, and when I reached out to them, they said, can you please come and talk about some of the newer Amazon services? Because one of the things they're interested in is some of the orchestration, which you might have heard about at this conference. It's called AWS Lambda orchestration, and also some other machine learning functionality that they're considering adding to this pipeline. But that being said, this pipeline is an example of an implementation of really the core serverless pattern, and they took a problem that was basically not solved by any other bioinformatician, applied just a standard serverless pattern. It's basically a static, it's a SPA website that has an API gateway that splits the target information, scores it, fans it out, uses an SNS topic, and uses DynamoDB for persistence. So also, in fact, Amazon has published this on Amazon's blog as a reference architecture. And this is not only useful for bioinformatics, like I said in the introduction to this. This is a serverless architecture in action, working, solving the problem. So started this out as a reference use case, and as I continue to work with them and augment it with newer Amazon services, if you're interested in this use case, you can follow CISRO's blog, my blog, or Amazon. So that's the first problem. That, however, isn't the only problem. A more difficult problem that CISRO is working on, that we're working with them on, is something Samantha's going to talk about.
A second problem genomic researchers face is that sometimes they don't know which specific gene is controlling the outcome that they're looking for. In this case, they need to look through the entire genome with genome-wide sequencing data association studies. These analyze large cohort data from reference genomes or imputed SMP array data, and use statistical clustering to try to figure out which section of the genome, which gene, if you will, is most likely producing the outcome that they look for, so that they know what to edit.
So in this particular case, as it says, they're viewing data sets with millions of features. Features are like columns for those of us that come out of the classic database world, so super wide data. So again, just making sure that we're understanding this, Samantha, this problem is entire groups, whereas the other problem is more individually targeted. Is that right?
Correct. Both of them are probabilistic, but this would be much more computationally intensive.
Yeah. So again, looking at the pipeline pattern here and kind of drilling in, because there's a whole bunch of things that are happening now with machine learning on the cloud. And this problem was particularly interesting to me because this problem of not only implementing but properly scaling machine learning algorithms on the cloud is really something that in my architecture practice I'm being asked to do very frequently now because of the volumes of data coming in, whether it's from bioinformatics or IoT or some other type of method, but just so much data is being produced that these very complex probabilistic cloud solutions is something that my customers are looking for me at least to try out with them. So this pattern that I'm seeing is when a customer is trying to figure out which machine learning algorithms are going to provide most value to them it's an iteration within an iteration or a set of iterations. So the iteration path tends to be you bring in the data, you process it. The processing for machine learning might be different than just regular adding up the data because you might do things like bucketize it, look for outliers, all the statistical analysis that you might want to do. And then often people have a data science bent will want to visualize the data. So including a visualization component or an ability to visualize during this iterative development process of a pipeline is something that is relatively new pattern for me in building out these big pipelines. Another thing that I'm seeing, and this is represented by the ad hoc query box, is this desire for multiple languages. Some data scientists use R, some use Python, some use some machine learning library. So the ability to kind of plug in languages into the test environment is something that I've had more and more demand for. So CISRO had a solution in place, or has a solution in place that they are using. They have an on-premise solution for doing these GWAS studies, and that is using Apache Hadoop. So they have a local Hadoop cluster, and the idea there is Hadoop is much more scalable for the size of data that they're working with. It's many, many, many terabytes of data. And they have also selected Apache Spark to make use of in-memory computation. We'll talk a little bit more about that in a minute. And then they have written a domain-specific open source library. When I met them in February, they were starting to experiment with placing this on the cloud. They really, again, like they did with GTScan, they are a research organization. They want to make their work more globally available, and it's their goal that bioinformaticians can collaborate as these data sets are gathered worldwide. So they were looking to leverage cloud pipeline patterns. So this is just a picture of what they have. Their library, their open source library, is called Variant Spark. It has two primary aspects to it. It is a domain-specific library for bioinformaticians, so the processing uses words that bioinformaticians are going to understand, like variant analysis, which is matching the genome sets to reference genomes, like the 1,000 genome database. And the secondary aspect, it has machine learning. This is in their corporate data center, and as you might expect, they're running into some challenges. If you've ever run a Hadoop cluster locally, it's resource intensive. Even though it's open source software, the expertise around Hadoop is hard to find, and it becomes very, very expensive very quickly. So, kind of an aside, I have done quite a bit of work on Hadoop, actually, just over the years. Done some training on it, so if you're just getting into it, you can just look me up on the web and have a number of resources available there. But a trend that I've found in working with Hadoop over the past 12 to 18 months is with the development of the Apache Spark library, which is designed to sit on top of Hadoop, although it can sit on top of other things like S3 or Azure Blobs, so on and so forth. You have this unified library that does distributed compute in memory of commodity machines. So what we had previously is we had a bunch of just disparate libraries that were really difficult to deal with in Hadoop. and as a trend in really huge data. And I think this is actually important for the Norwegian market because as I've researched, you guys have a lot of oil and gas, sensors, industrial IoT, and I've done some work on that domain as well. Those volumes of data warrant Hadoop. And what's happening with the Hadoop ecosystem with Spark is a maturity of the technology. Spark, interestingly, well, you know, where was Spark developed?
UC Berkeley in the United States.
And who developed it?
A team at UC Berkeley.
Yeah. It was a group of bioinformaticians, basically, to solve this problem. So Matea, I forget his last name, he is actually the guy who came up with Spark. What's interesting about it is if you look across the top here, you can see that you've got Spark SQL, Spark Streaming, Spark GraphX, MLlib, and all these languages. And this is how a bioinformatician, or more broadly, a data scientist thinks. This is something that, I want to bring trends to you because I know you're developers and you may not be working in bioinformatics. When you get into true big data and you work with data scientists, they really are not usually married to one programming language. They will use multiple programming languages, and I find that to be very different than application development I've done over the years. So kind of an important meta takeaway thing is if you're working in big data, it's time to learn Spark. It's really an important technology, and it's bringing Hadoop of age, and it's used in this solution. So not only did they use Spark, but they used Variant Spark. So what is Variant Spark again, Samantha?
Variant Spark is an open source library that allows for the processing of data from studies like Genomic-wide data association studies in an incredibly fast, sophisticated manner.
Yeah, so when I was at CISRO, after I talked to them about some of the new Amazon technologies for GT-Scan 2, I said, hey, what else are you guys working on? What else could you use the Cloud Architect for? And they said, well, we have this Variant Spark, and we think it's pretty great, but we really haven't had much pickup in the bioinformatics community. And one of the things I said, do you have a reference use case? And they said, well, yeah, we published a study. And I said, how do you get it? And they said, well, you go to GitHub. Now, this is something Samantha and I actually have quite a lot of experience in, but from a different domain. Separately from our professional work, we run a nonprofit called Teaching Kids Programming, and that is an open source library. And our target for that is school teachers. So one of the lessons we learned in working with that project is that people who are not developers, they don't go to GitHub, do they?
Not at all.
They really don't. So for Teaching Kids Programming, we actually got a grant from Microsoft, and we have a website, and everything is indexed, and it really increased the uptake. So kind of a high-level lesson about using open source libraries in any pipeline and getting sort of an uptake on it is to create a reference example. And so that's one of the things that we've been working with CISRO on. We're going to kind of move towards that. But before we do that, we want to talk about why Variant Spark is so interesting. So to that, Samantha's going to talk about it.
By means of comparison, Variant Spark tested the speed of their application versus other common sources of statistical reference. And this paper was published in 2015. They found that Variant Spark, tested on the same data, was 80% faster than Atom, a similar service from Berkeley, and 90% faster than both R and Python.
Yeah, so it's interesting. So benchmarking is really, really important, and frankly, benchmarking is some of the work that we're looking to do with CISRO. So if any of you guys want to join the party here, I love to benchmark big data pipelines. I think it's just a really fun thing to do. So you can just contact me after, because I know it's a big learning for me whenever I get that opportunity, because in fact, the underlying architecture of Variant Spark has basically improved since this benchmark was done. So Variant Spark is this open source library. It's built on top of Apache Spark. Specifically, it's an extension of Spark ML. The idea is to massively parallelize the generation using the random forest machine learning algorithm, which is sets of decision trees, to identify genes efficiently. And Samantha, you want to talk about the results that they got on their benchmark?
This is a landmark system. It can analyze up to 3,000 samples with 80 million features in less than 30 minutes. Now remember, samples include the entire human genome. This could enable real-time diagnosis of patients with diseases like cancer by finding similar patients in the reference genomes. In fact, it's already in use in Australian medical research. It contributes to motor neuron disease research, and, well, you guys might know this as ALS, popularized by the Ice Bucket Challenge.
Yeah, so it's a really exciting library, and again, one of the reasons that I chose it as the subject of my talk is because there are some other competing libraries out there that they're also good. But Variant Spark has got some really impressive results, and I really would like to see more bioinformaticians. So if you work in a medical facility here in Norway, tell your bioinformatician friends to try it out, because the results are really pretty darn impressive using the modern big data technologies. Now, I could do a whole talk on machine learning. As you guys, we're all learning machine learning, because if you haven't used statistics since college or whenever you learned it, it's a lot to relearn. And just as an aside, I'll tell you, I am personally relearning statistics, and I'm finding it to be very, very useful. The process for creating successful machine learning sections of data pipelines is really complicated and hard. And the things that I'm finding that make you have success around it might be surprising to you. It's all about understanding. It goes back to thinking about problem, maybe you've seen it in a different way, where you call the 10x programmer. You don't want to have the 10x statistician. In other words, if you have the one person who can implement machine learning and nobody else can understand it, that is definitely not the way you want to do it. So the problem is around education. And I see this again and again with customers. I see machine learning algorithms that the one person implemented. When that person leaves or when there's some sort of a problem, then the whole pipeline falls down. I worked with, just as an aside, a YouTube remonetizer that had millions of dollars in startup money, and they completely folded because of the 10x data scientist. So how do you get around this? You involve a team, you work together, you go through these iterative cycles with data prep, statistics, probabilistic algorithms, and most importantly, data visualization. You want to create a vehicle or a workspace, or in our case, it'll be a Jupyter notebook, where the team can understand what's happening together. So to drill in a little bit, Samantha, do you want to talk about this?
Actually, I had something to add about machine learning,
Oh, sure.
if that's all right. This is imperative to the bioinformatics community in terms of research. We want to be able to use these tools the first time, and we want to be able to repeat their use again in different communities who have never heard of it before. So this process of making it accessible to multiple people, regardless of their interest in code or statistics, is absolutely imperative.
Yeah, it's true. And just one more thing. I read a study that 70% of the published studies that they examined for this particular study, the research was not reproducible. And I was stunned because of lack of technical, sophistication. And I was just stunned. So I did some other work with Docker just to help some people on that. But anyway, so coming back to this machine learning. So Variant Spark uses wide random forest ensemble of decision trees. And there are other libraries out there that use things like logistic regression, which to you right now probably means nothing. But we're going to show you through an example in our notebook of how this can be visualized so researchers can understand the differences. So, working first with the library before we get to the notebook. These are common best practices around a library. When you look at a library, and I'm actually working with a team right now to clean and work with the library, you want to make sure, is it usable? Is it performant? So we need to revalidate the benchmarking. Is it extendable? I found a whole bunch of commented out code, typical kind of stuff. You see a lot of this, especially coming out of research, out of universities. The folks that are doing research, they have never really had any discipline around coding practices, and so maybe they're not even doing unit tests. They have all kinds of comments, all kinds of code paths that should be deleted. So you want to have the rigor when you're bringing this research code into production quality. Are they using the best language? Again, this is all about understandability, reproducibility. The library is written in Scala. I've never done anything in Scala before, so I'm using myself as a guinea pig, and I'm not finding it to be the easiest language. I do most of my prototyping work in Python, and I've actually been talking to the people who make Spark about what the performance difference would be if we just switched it to Python, because it would certainly be a lot more understandable by a greater community. Or should we have it in both? Are they using the best version of Spark? I found that this library is on Spark 1X, and they have machine learning running on top of it Spark has gone to 2x, and there's a whole bunch of improvements in Spark ML. So before we benchmark test it, we have to follow clean coding processes. And again, the pattern here is if you are writing one or more modules, particularly if they have machine learning, you have to have the software disciplines that you know and love from work already applied to whatever modules you're working with. And then, is this machine learning algorithm understandable? This is a problem we're really wrestling with. All right, another challenge for this particular solution was how best to deploy Cloud Hadoop. So we have really three options. We could do infrastructure, platform, or software as a service. And I recommended to the team that they go straight to software as a service because they're a very small team. They're having a very difficult time finding technical talent who also know bioinformatics. They need their bioinformaticians to focus on the bioinformatics problems. And in the case of Hadoop with Spark, there's actually a vendor that I've worked with quite frequently called Databricks. Databricks is the commercial home of the people who make Spark. They basically made a company and they add services around Spark. And in the US where I'm working, they're basically on fire. They actually have conferences, and I don't work for them. I just think it's a really fantastic offering for these domain-specific use cases. They abstract away. Right now they run on Amazon, so you're running on EC2, optimized instances, and they provide enterprise security and other services that, particularly in bioinformatics, are going to be needed, and let the bioinformaticians kind of get to their job. So one of the great things that they provide is a custom implementation of a new type of workspace called a Jupyter Notebook. Now, there was actually a talk at this conference that I recommend you watch on Jupyter Notebooks. Jupyter is an open source technology. The implementation in Databricks is their custom version of it, and it's something that you can try out. So I'm going to actually show you how to try it out here for up to 1 hour for free using the community edition. So to start, you just click Create Cluster, and you have a 6 gigabyte memory cluster of Hadoop with Spark on it, and they have a whole bunch of reference Jupyter Notebooks. So the Jupyter Notebook is a website that has multiple sections that you can plug into it. One section is going to allow you to do documentation. It's basically Markdown. And so you can think of it like a wiki here. If I click in, you can see it's Markdown. What's more important is for the runtimes that are associated to where this thing is running, and this notebook is running on an Amazon EC2 machine, happens to be in the US, on a cluster, I can run, you can see up here by default, it's Python. So if I click right here, I'm basically checking that I have Spark on this cluster, and you can see I have Spark. So this notebook is a demonstration of an aspect of the API. And for you who work in VS Code or Visual Studio or Eclipse, you can think of it almost like a new IDE, a different kind of IDE, a data science IDE. So you can see that here we're going to load some sample data, and I'm just going to go ahead and click Run. And what this is an interface to the Spark cluster running, in this case, on a single machine. And then this just goes on, and I can go ahead and I can do processing. And then I can also use other languages that are installed on the cluster. You might remember from the Spark slide that there is Spark SQL, and this is similar to ANSI SQL. In addition to just running a SQL query, and hopefully you can see this, okay, you can also graph. And this is so important to the data science workflow. So this is a general notebook. This is something that you get with Databricks. Oh, also inside of here you have revision history. So you can check it into GitHub. You can put comments. You can permission it. It's a fantastic, not only demo of variant Spark rather than a GitHub repo, it is also a reference way for bioinformaticians to work together. I think that when Samantha starts college up in the fall, she's going to be using these day in and day out in her research as a bioinformatician. And we as a developer group, we have a sort of a new interface to consider when we're working with these big data solutions. And that's why one of the reasons I wanted to show this to you. So that's kind of how these work in general, but we're going to have Samantha show how this works with Databricks. So if you want to switch. So go ahead.
Now, we all know that this technology can be used to solve very important questions, and as we said before, revolutionize the way we treat diseases like cancer. However, much of that patient data cannot be released for privacy reasons. So, the team at CISRO got separate data, equally real, but for a little less dire of a problem. Genetically, who might become a hipster? If you have yet to see a hipster, here are some reference images. There's one. There's another.
We weren't really sure. I went over to Mark Vian, I think it is, and I spotted some people like this in Grita Loca. Is that where you go to find hipsters around here? We checked it out. So yeah, so we're gonna actually show you, and you can switch to the notebook, Alt tab. We're actually gonna show you a working notebook that doesn't look for cancer. It looks for the genes for a hipster. This one actually. And academic, yeah, right there. And variant spark hipster. So go ahead, Samantha.
Now, we can't summarize every single aspect of a hipster. After all, they are people and they are different. But we found 4 distinct traits, or the team at CISRO found 4 distinct traits that they wanted to aggregate and look through the genome for specific indicators that these traits might develop. These are the growth of a monobrow.
Monobrow. Is that translating monobrow? Okay.
The development of certain horizontal cells in the retina that indicate a preference for checked shirts. The growth of a beard, and a high preference for coffee.
And Samantha, you can see they're on. They've cited the genes and they've cited the genomic reference papers. So these are actual genes that correlate. This is published research. Not kidding. Okay, so that's the problem. So if you want to scroll down a little bit, if you want to click over to the cluster over there, the cluster, yeah. So I'm on an academic shard here, and you can see if you click right there that I've got 6 nodes. This is just a demo cluster, and then if you go to libraries, you can see that I have variant spark, which I've just uploaded. Click on variant spark. It's a jar file. So you just upload that, and then go back over there. You are going to go, and you're going to go to the attach, right there, yeah. And then you attach this notebook to the cluster. So it's your UI for the cluster. And again, it's a fun example, great for this use case, but also from a trend standpoint, if you go to a data science conference, like this is what the data scientists use. They don't use VS Code, they don't use Visual Studio, they use Jupyter Notebooks. Also, these are renderable in GitHub. Like they will show. So, I mean, just as a general trend, you can install them locally too, just to play around with notebooks. There's also a commercial vendor called Beaker Notebooks that has all the runtimes associated, and I used it to learn like Julia and some of the newer languages. So very important notebooks. Okay, let's get to the task at hand. So what's the first step there? Step 0, what do we have to do?
You would download the Variant Spark libraries to your computer.
We did that, yep.
We would import the library into Databricks.
We did that.
We'd create a cluster.
Yep.
And then attach the notebook to that cluster.
Great. Okay, so now we're ready to run our analysis and find out about the hipsterdom here, okay? So what's this first section, Samantha?
This is the part where you'd load the data that you'd collected. So this is the part where you'd load up your samples and try to figure out from there which genes you were looking for.
And this would be, can you go ahead and run that? And they're using Python there. Notice the % Python, because it was just more convenient for them, and because this is a Scala notebook, because Variant Spark's written in Scala, so you can see on line 1 on that little section there, because they wanted to use the URL libraries, and they're loading this from S3, and this is VCF data, and where does that come from?
Set of reference genomes?
Yeah, the reference genomes and the sequencer. That's the data off the sequencers. So, okay, so this is our samples, our data. So now what's our next step?
You would load the variants that you already had based on your references.
Okay, cool. So let's run that. Because we're doing matching to a reference genome. So you can see, like in lines 1, 2, and 3, we're calling the variant Spark API. Basically, we're instantiating it. And then we're calling on line 5 on the VX context, which is the variant Spark context rather than the Spark context, like the database connection, the feature source, and we're bringing in the samples. Okay, and now what's happening on 3 there, Samantha?
We would be loading our labels, or basically the headers in the Excel columns that we were looking for.
Yeah, and why do we have to have labels on our data in this for this situation here?
Well, the human genome, of course, has so many different features that we could possibly look for that here we want to specify and make very sure that the algorithm only needs to look through what it has to.
Yeah, and it's a supervised machine learning algorithm, so we provide it with labeled or known data versus unsupervised, where it's like clustering, where you just group the data together. So it's known, labeled data, and then a set of algorithms, right? Okay, then what's next?
Run it through variant Spark.
So we got the analysis. Yeah, that's our import analysis, and then we're going to run our analysis. Okay, and this is our big computationally intensive thing. So this is our set of decision trees. So we're breaking it down and we're matching, and we're iterating over groups of trees to find the best matches because it finds correlations, right, between the gene types. So whether it's the monobrow and the beard, or the monobrow and the check shirt, or which things are most closely correlated. What do you think it's going to be? It's going to be hairy, caffeine drinking. We'll see. So this takes about 4 minutes, and this is an area where CISRO came to me and said, we want to make sure that what we've written that runs on our local cluster, because this is the intense machine learning part, is optimized properly for the cloud. So what we've done, and if you switch over to the repo over here, I actually forked this repo. So again, I love, I'm a lifelong learner, and so I love to collaborate with people. So if I'm collaborating with some university students who just finished data science, and we are refactoring CISRO's decision trees and k-means algorithms, which is what they first started with. So if anybody wants to party on this project, we'll be back in the States, so it'll be over Skype. What we want to do is refactor this out, and then we want to start looking at some of the high-performance deep learning libraries that Amazon and Google have supported. They're open source, so MXNet and TensorFlow. We've been doing some work with TensorFlow, just trying to see if that's going to be a better fit, because interestingly, Spark, the Spark people announced at Spark Summit that their core object is a data frame. That's how they parse the data. That they have now made something called a tensor frame, which is optimized to work with TensorFlow. So to me, this is the real interesting problem space here, so long as we make sure the algorithm is the correct algorithm, which it seems to be, how we then optimize it. So this is my fork, and you can just send me an email or something, or fork it yourself if you want to play with it. So how are we doing on this? Is it running? 500, okay, still running. So the result of this is we're going to see which of the, what are they called, phenotypes that are most associated?
Which of the genes specifically has the most impact on the development of all the hipster characteristics. And I think this is the results table already loaded.
Yeah, we already loaded it. So now let's visualize it. So this is running SQL, and this is, like I'm saying, this is a reason for using this notebook interface, because we can use SQL. So they're dying to know what's the top result.
Well, it looks like here that the gene with the most statistical importance in the development of these hipster characteristics is the one for the development of a monobrow.
So look in the mirror. We got some hipsters in the room? I don't know. So again, the greater point is, in this interactive notebook, the data scientists can work in SQL, or if you scroll down, you can see there they're visualizing in Python with, what's with that? It's ggplot? Pandas, Python with pandas, which you did that at Stanford, didn't you?
Yeah, for statistics.
Was it fun?
It was not.
Anyway, scroll down. So maybe R was more fun.
Yeah, you could say that.
Okay, so scroll down. We have the same thing in R. Scroll down. Same thing in R. Scroll down. And the fun people at CISRO actually made a infographic because they were pleased with their results, and you can see it truly is the monobrow. So another aspect of working in this collaborative environment that is great for bioinformatics is the ability to do alternate analysis. And they actually did this. So if you scroll down a little bit, can you talk a little bit about that?
Sure. So a competing service to this one right here comes from Hail, which is kind of the same thing, but they calculate the importance of certain genes using a statistical method called logistic regression. And you'll notice in their analysis right here that...
Probably just going on to the plot. It's probably easier to see.
Yeah. They did not include the monobrow gene as most statistically important. They got a different result using this statistical method.
Which may seem humorous, but it's kind of scary in a way. And it goes to my greater point of the importance of understandability of machine learning, because these algorithms are complex and there are relatively few people who can explain them. So a very big area of opportunity in this world of big data is the proper implementation, visualization, documentation, and explanation of machine learning. And it's an area that I intend to be working in over the next 12 to 18 months with the bioinformatics community. So if you're interested, follow my work. I'll probably do some IoT stuff too, because just have work in that area. But it's just fascinating to me that we can use two different bioinformatics libraries and get two different results. So let's switch back. All right, so a couple more things about making the decision for using software as a service for this particular use case. This conference has been a lot about serverless. It's been a lot about serverless basically. And I don't want to go away from some of the other patterns that work. In this particular case, not only does Databricks abstract away all of the Amazon implementation details, they're also really smart about working with Amazon. So in this case, it's just a matter of using the slider to use EC2 spot instances. And this was actually a real problem that CISRO was encountering when I talked to them. They said, well, we know spot is out there, but we don't really have somebody to sit there and bid on spot and see when spot kicks up and that kind of stuff. And if you're not familiar, this is just unused compute. And it's a very typical use case when you have these big and variable computationally intensive workloads that you just bid some really low price on a spot instance and you can make the workload go a lot faster. I mean, just to kind of bring it to the real world, one of the runs that they were doing on-prem, I think a one run to get one result was 55 hours. And I walked away and I said, I'm doing near real-time ads for some social media service in LA, and I want to bring those technologies to problem spaces that really matter. I don't want bioinformaticians to have to wait 55 hours every time they run something. So I'm going to take techniques that I've learned in other verticals, and I'm going to help the bioinformatics community to use these techniques, whether it's through vendors or other processes. So this is the solution as it stands right now, the recommended implementation. So it's really simple. They're using S3 buckets. And they basically have software as a service on this, basically big cluster, and they have the spot instances. And the idea is that this will be able to scale out and be easily replicatable for bioinformatics communities worldwide. So in terms of the pipeline pattern, what we did for this analysis is we selected S3 and Hadoop. For the ingest, it was S3, and then into Databricks file system. For the ETL, it was Spark. For the analysis, it was Variant, Spark ML. For the visualization, it was notebooks with SQL, R, and Python, and it was a software as a service recommendation. Now, just to kind of tie it back together, because there are two different problems, two different patterns, you'll remember the other GT scan, the Google for genomics, if you will, sort of. That was a pure serverless implementation, working well, scaling, just working perfectly. And the way that was working out was you're going to use at the top level there for the VCF files S3 and Dynamo, for the ingest S3, pretty much everything else in Lambda and API gateway. So for some of these big data problems, serverless has been the right answer. For others, something that hasn't been much talked about in this conference, software as a service. And there's many, many other software as a service implementations. Another one, I'll just throw it out there that I've been doing some work around is algorithmia, which is machine learning algorithms as a service. So there's lots of stuff out there to look at. So that's pretty much what we have. So I'm Lynn Langit, and hopefully you found this useful. And
I'm Samantha Langit.
Thank you very much. we do have a couple of minutes, so if you have questions, I cannot see anything, so you're going to have to really yell out. We'd be happy to take questions. Are you guys just weary at the end of the conference? I appreciate you coming out. Okay, well, if there's no questions, thank you again. Thank you.